next talk, what I wanted to share with you is actually something that we do in medical services already. This is not just research, but actually operating in a clinical setting. There's a numerous companies that do this, and what I wanted to do is to do some kind of a technical comparison of why you do this one way or the other. And I want to start this talk by a bird's view on clinical and pharmaceutical drug development. And what this graph from Nature Reviews on Drug Discovery shows us is an evolution of the pharmaceutical companies' success rate. And in this y-axis, what you see is reasons for failure of new drugs. And on the x-axis, you see different reasons for why drugs could potentially fail. And what we can see is that some reasons for failure of drugs, such as pharmacokinetics, meaning whether the drug is actually available and lasts and stays in the body of the patient and reaches its clinical target, are actually reasons that stop to exist between, in blue, 1999, and in gray, the year 2000. But other reasons continue to linger by the year 2000. And really, if there was in this graph the year 2015, what you would see is a significant decay of this graph here with efficacy. And the reasons for this is what I will share with you now. I already talked to you today about how therapeutic window is actually subject to a normal distribution. And much of the distinctions between patients that respond and do not respond all derive from the discovery that within cancer, alongside the diverging morphology of the tissue, there is also significant and systematic difference in the sequence and gene expression, sequence of genes and the gene expression, as well as obviously aneuploidy that was discussed before. And this ultimately allows us to distinguish between patient groups of treatment. What we are trying to do is to somehow, in a broader sense, summarize overall what is happening in a patient and try to derive from this therapeutic regimen. So, for instance, what Raj Geyer, a PhD student of mine, has done to these 238 glioblastoma patients is to look for recurrent occurrence of a mutation or a lesion or some genetic lesion in one or any of members of the same pathway. So if you have the cell cycle regulating pathway that is centered around the RB and E2F, then what you can see is that approximately 4 genes are always, one or the other of them will have a mutation in a glioblastoma case. The same for p53 pathway. One or the other of 4 genes will always be aberrant in a glioblastoma, and the same for the MAP kinase, tyrosine kinase receptor pathways. Always one of those or the other will have a mutation in it, but specifically only this group here, within the p53 pathway, only this group here will actually respond to a new drug called natlin-3, which causes the proteasomal decay of p53 wild type. So if the mutation is either downstream from p53 or on p53 itself, then there will be no response to accumulation of that p53. But if the p53 is wild type but undergoing proteasomal decay, then this patient will respond to a drug called natlin-3, and we have actual patients like this that we treated based on this rationale and got responses. Similarly here, only if the EGF receptor mutation is one that activates the EGF receptor itself, you can expect EGF receptor inhibitors to work, etc. So basically, from analyzing different activities in cancer from proliferation, but as well as evasion of cell death, etc. We're trying to develop these types of network or circuitry analysis that ultimately should deliver a decision how to treat this patient with targeted drugs. And much of this is centered around this machine that allows the sequencing of approximately 200 genomes within 6 days. And just briefly how this works is that if you have some sequence of unknown nature, all you need to do is to attach to its edges known sequence, which is printed on surface, thus the molecule of unknown nature will get stuck somewhere. Then through bridged PCR, you get accumulated spots. And essentially, we can analyze a few billion spots like these at once. And each one of those spots will, alongside or along time axis, will change its color depending on which of the dideoxy bases incorporates in each cycle. And if you track the same spot by its address on the surface, you can see that it begins with green, blue, red, etc. until you, and these green, blue, red signals accumulate into bases, and that's what we analyze to discover new genes that occur in cancer. When we did this, originally there was and I'm talking 16 years ago, and a huge increase in our capacity to do that at around 8 years ago. We were really expecting that we will find new genes and new ways to treat cancer. But what we discovered was that the frequent mutations in cancer were already known. And most of the new mutations that we found were actually exponentially rare. And this makes the medical management of cases based on these cumulatively rare incidents more and more difficult. That's why, in order to make this feasible as a clinical management tool, we need to actually analyze the meaning of each of these rare mutations and see where in the overall scheme of activities this particular mutation builds into, and try and summarize this into more simplified versions of whether this cancer is more sensitive to mismatch repair or homologous end joining, etc. And you've seen this graph already that illustrates the clinical benefit of finding patients that will respond to a drug. Essentially, most of the patients arrive to us after they've been treated with guideline-based, evidence-based medicine, treated, and somewhere here, options run out. However, in addition to those, there are also cancer patients which on the outset we can recognize that the guideline is not relevant to them. Specifically here, shown ovarian cancer patients, stage 3 serous papillary patients, which through 3 different sets of biomarkers, either amplification of cyclin E or overexpression of extracellular matrix genes, or through increased translocations, numerous translocations across their genome, we know that these patients won't live through a year if they're treated through the guidelines. And they represent approximately 1/4 of the ovarian cancer patients. If only 35% of the ovarian cancer patients die within 5 years, 25% of overall ovarian cancer patients die within 1 year. So this 1/4 of the patients should be looked for alternatives in the outset. And the alternatives could come from clinical trials like this, where Ben Solomon, together with Grant MacArthur, participate in the introduction of a new drug called crizotinib. And crizotinib is an inhibitor of the TGF-beta receptor ALK gene. The amazing thing about this and the relevance to the discussion is that only 1 in 50 lung cancer patients actually has this mutation. So if you don't know how to recognize these 1 in 50 patients likely to respond, you won't be getting this amazing benefit from these drugs. So basically, a modern oncologist is perceived as as a matchmaker, where he has a large arsenal of arsenal of targeted therapies, and he needs to use genomic technologies in order to matchmake between which drug to which patient. Basically, you take a tumor sample, you do the sequencing. The sequence analysis means that you take those 500 or 200 to 500 pieces, find where they belong in the genome, and find what's different about them from the normal tissue of that same person. And this needs to be ultimately converging to a recommendation. Of course, we don't want to read the whole genome because only a small fraction of the genome is meaningful to us. And the reason to focus only on the region of the genome that you're interested in is because within the same run, you can read many times the same base. And the more times you read the same base, the more clinically relevant this analysis will be, because you'll be able to see rarer and rarer subpopulations within the cancer, which is related to the previous talk. So eventually, you have to choose where do you read. And because so far, internationally, the International Cancer Genome Consortium and projects their likes have analyzed approximately 20,000 cancer genomes so far, if you have this information, you can exploit it to only analyze genes that are appearing in the population within a certain frequency with mutations, and that these mutations within the person that carries them represent a large fraction of the cells, thus indicating that it's a driver mutation, etc. What I would like to share with you is extensive analysis that we've done both in the University of Melbourne. This is Sebastian Lunke, who runs the Royal Melbourne Hospital molecular pathology labs, and Roy Feingersh in Tzfat. And together, we've analyzed about 7 different ways how to restrict the region in the genome that we're analyzing to what we think is really relevant. The first step, as I said before, is to introduce the edges which we know, which will allow this piece of DNA to stick to the surface. So there are different ways to do this. One is to use transposon, a variation of transposons. Most of the methods, however, use sonication to break the DNA into pieces and then attach to the edges, enzymatically attach to the edges the sequences that are necessary. And otherwise, there is also an option of using restriction enzymes. The problem with restriction enzymes, if you use restriction enzymes, is that the first base you will read will always be the same. And the same problem applies to PCR. So the advantage of using sonication or fragmentation is that the edges of the sequence are random, and then the next step is to capture the region, and there are many ways to do that as well. All of them are relying on magnetic beads which recognize biotinylated nucleic acid, just that whether the biotinylated nucleic acid is hybridizing and complementary to the sticky ends of restriction sites, or like a PCR primer, or is it inside the sequence that you're interested. This will make a huge difference, because let's focus on the last 2 options or 3 options. If you capture the sequence you're interested from within, and then remember your sequencing is coming from the edges, this means that you don't actually need to know the full integrity of this piece of DNA. So imagine a scenario where there is a gene called BCR and a gene called ABL, or TEMPRESS and ERG, and those 2 have fused in the cancer genome. So if I'm based on the edges, such as restriction sites or PCR, then I only can analyze the wild-type BCR and the wild-type ABL. But if I'm not relying on the edges, a hybridization element in the middle of BCR may pick up a piece whose edge is coming from ABL. So when I will map this sequence, I will see that it doesn't belong to where my capture was, and this is how I can identify fusions, and because the cancer driver mutations fall in different types, not only base pair substitutions, but also copy number variations and translocations, you want a method that will be able to capture all of these things, and you want a method that if you can see an elevated representation in your sequence, this means that there is an amplification. So that's a problem when the edge of the sequence is always the same base. You can't be sure whether this is an amplification bias or a true amplification in the genome. So, okay, so you choose your genes of interest, and then you try to qualify it. And similar to the previous talk, the first thing you need to do is to dilute DNA which contains a particular mutation within a DNA that does not, and see where your detection limits are. And in this case, what we find is that the best we could get to is about 1%. We're now working on a new method which we believe will bring us down to 0.1%. A point just a comment about this is that because the DNA we're analyzing is formalin fixed material and often comes from the pathology labs from a few years ago, this is really a limit, a lower limit of our detection, because at the end of the day, in the formalin there are all sorts of things that happen to nucleotides which are not the mutations in the cancer tissue originally. So you can't really go down below that. Then you want to know whether, if a method is very effective, then increasing the number of reads will give you an increase in the number of mutation detections. And some methods like rain dance do not do that, and that's originally excluded. Then you want to see that you can recognize all different forms of mutations, whether they're missense, indels, etc. And then you want to know if, for a known panel of cancers, if they carry a specific mutation that was detected by a standard methodology, you want to know that you never miss one of those, and that's the answer for at least 5 of those methods that I described to you. And you want to know that if you find a mutation, so you want to know that you're not missing, there's no false negatives, you're not missing things that are there, and you want to know that if you find something, that it's truly there. And lastly, you want to know that you can reproduce the same result time and time again. So basically, in the end of the day, it boils down to cases which are ultimately untreatable, and then they come to you. You find new mutations that obviously a significant part of hindrance to this methodology is interpreting the results. Then you give a treatment recommendation, and you can see if the patient responds or not. And basically, each story to itself, and this is really one of the rate limiting steps of the progress in this methodology, is that you cannot, it's very hard to collect many different cases which follow the same story. Each case it has its own history, medical history, physiological situation of the patient. Your recommendation is divergent, and therefore it's very hard to do evidence-based medicine in this approach. And lastly, one of the bottlenecks in this methodology is simply how to interpret new base mutations. So there are some mutations like the amino acid 848 of EGF receptor, or amino acid 600 of BRAF, amino acid 12 of KRAS, which are clinically recognized and everybody knows what to do with them. But what happens when the amino acid you found was one above or below this amino acid? We don't really know what to make of those. So this is really why we established what I talked about in the previous talk, which is this methodology of avatars, of growing the tumors in mice. Originally, the purpose of it was to decipher the genomic, the clinical significance of genomic aberrations of unknown significance. And just as an example, I'm bringing 3 such cases where We found new alleles that represent sensitivity to PARP inhibitors, and some of them are unexpected and don't even belong to the BRCA1 and 2 complex. We found new alleles that represent immunological checkpoint inhibitor sensitivities, and some of them are not expected, nothing to do with the PD-1, PD-L1 match. And we found new alleles for kinase inhibitors, and some of them are not expected. So overall, so far, we sequenced about 500 cases. Just for reference, Foundation Medicine, which is the leading company in the world for this, has already sequenced 30,000 cases. And yes, larger is better in this field. But what we have done in addition is introduced 150 cases into transplanted CDXs, and because we analyze the responses, we can generate interpretation or curation, which is novel and unique to us. And this leaves me with just thanking you for your attention.
Questions? So you mentioned in pathway analysis that the proteasomal activity has increased. So did you look at the MDM2 also? Yeah.
No. There what?
There was an increased proteasomal activity.
Yes.
So do you look at the MDM2?
Yeah, no, that was these were cases with MDM2 amplifications. If they have MDM2 amplifications, those cases will be responding to NATLin-3 because their P53 gene will be wild type. Yeah. Thank you.
Questions? I have one question.
Sure.
So in the cases where you find mutations that are more rare, do you look at whether more frequent mutations are going and would you then potentially discard the lower frequency mutations, or would you still pursue them?
There is a common denominator of these a rare mutation. Mutation can be rare in two ways, which is confusing. One is within the patient. There's a tumor. I'm reading the same base 10,000 times, so I find it 5 times out of 10,000. If I don't know for a fact that this is representing a resistance to a particular drug, and after treatment this will take over the whole tumor, I disregard that. And there is rare because it's rare in the population. So I read 500 individuals and I only hit it once. Mostly with these, they represent a gene that is a modifier of a known pathway, which is what I try to show with these glioblastoma cases. So even though it's appearing only in 1% of the patients, it really represents a notch pathway failure or a PI3 kinase activation pathway, etc. So for all practical purposes, in terms of drug recommendation, it's still the same PI3 kinase.
That's
I would recommend.